I'm very grateful to have the graveyard shift here. But we do have some very interesting talks, funnily enough, and some of them I know something about, which is even better. I'm Mike Eaton from the ETP and Nanomedicine, and we have a very good lineup this afternoon, started by Cristianne, who's going to take us through her near phase 1 study. Is that correct? So I'll say no more. What I'm going to do is 15 minutes talk, and we'll have 5 minutes immediately afterwards, in case people vanish off before the end.
Yes, good afternoon, and thanks for staying so late, and also thank the organizers for being able to present Cristal Therapeutics to you. And actually, the day before yesterday, it appears that I'm following, or actually having, one of the 4 essential characteristics of being in life science, namely that I'm a little bit optimistic. And why is that? Because as you'll see in the overview and the abstract, it says phase 1 study. Unfortunately, we do have the ethical committee approval, we do have the material available, but we not yet started off with the phase 1 study, so that's why I had to put a word towards a phase 1 study in the title of this presentation. But I'm more than pleased to show you what we're working on and why we think we do have a good product for patients with advanced solid tumors. So Cristal Therapeutics is a pharmaceutical company developing innovative nanomedicines. We are officially a startup of the Utrecht University, but since 1.5 years we're nowadays located in Maastricht, which is the south of the Netherlands. We're venture capital funded, and approximately 10 people are working for us at the moment. So using CriPec nanomedicines aims to improve the therapeutic performance in a stepwise approach, namely by initially assuring the stability of nanomedicines in the bloodstream, having a really long circulation profile, thereby improving the disposition of the nanoparticles over the body, targeting the diseased areas, and having an improved accumulation at the target site as compared to the native drug molecule. But of course, once the nanoparticles have accumulated at the target sites, the drug should be released in a controllable manner. So how do we do that? We do that via CriPec, so that's the proprietary platform, Cristal Therapeutics Polymer Technologies. And the way how it works is a 2-step approach to generate nanoparticles from a drug in a 2-step approach into nanomedicines. Whereby the first step is taking the drug molecule, derivatizing it via linker molecule, so you really make a covalent linkage between the two. And then if you combine it with a proprietary polymer, you get a nanoparticulate structure form, in which, and that's different than as compared, for example, the liposomes that we've seen so far, or traditional micelles. In this case, everything, so both the polymer as well as the drug, are covalently linked to one another, as indicated by the gray bridges. So why is that essential to us? Because in this case, you really create one big macromolecule, so you assure the initial stability, allowing the nanoparticle to give it a different disposition over the body as compared to the native drug molecule. But what is essential at the same time is that in time, only the native drug molecule is released over time as a consequence of chemical hydrolysis of the drug linker bond. As well as that the nanoparticle itself also degrades as a result of chemical hydrolysis, also in this case of ester bonds hydrolysis. And by using different type of linkers, you can have different type of release profiles, few days up to several weeks. So it's a really tunable system in that regard. And what we've worked on last years is really optimizing the overall platform, demonstrating that the nanoparticles themselves are biodegradable and fully safe, that we can really tune the particle size as small as 30 nanometers. And this is an example of nanoparticles loaded with docetaxel. So as you see, you can go as low as 35 nanometers, but as high as up to 90 nanometers. And every time the polydispersity is really very low. So the way how we control the particle size is simply by using different type of polymer lengths, as indicated on the y-axis. So this really gives us the tools to control the particle size, but more importantly also, we were capable of entrapping a whole range of drug molecules in there, namely small molecules, paclitaxel, docetaxel, dexamethasone, so the corticosteroid compounds, as well as also peptides, shorter ones, the leuprolides, but also long-chain peptides, for example, as the exenatide, so a 39 amino acid compound, in which we, in all the cases, were capable of reaching a high entrapment efficiency, and to give you some values, more than 60% in case of the peptides, or in case of the small molecules, small hydrophobic molecules, up to more than 95%. And the only necessity in this case is to have a reactive moiety present for entrapment in the nanoparticle. So as indicated, depending on the indication and depending on the application, you have a different type of linker used to control the drug release profile. We have, and maybe other parties get as well, often the question, well, how do you position yourselves versus the other nanomedicine platforms that are being developed? And I do think we have an added value, or have a competitive added value in three terms. The first terms being in view of efficacy. We do have demonstrated improved or even superior efficacy in different disease models, in oncology as well as in arthritis models. I'll show you that we have demonstrated complete tumor regression upon a single administration, all as a result of the enhanced accumulation of the nanoparticles at the site of action, while at the same time, and that's the second pillar, having also an improved tolerability profile of the nanoparticles. And the third pillar, also not an important element, is the easy way of manufacturing of these nanoparticles. They are self-assembled. We've already successfully upscaled the production. So we have produced the first clinical trial batch, and the most important part is that the production process is generic, so also allowing the fast-forward development of novel products based on the same platform. So what we do ourselves is we're working on CriPec Docetaxel as the lead candidate product that is now about to enter the phase 1 study. We have proven the efficacy of CriPec dexamethasone in an arthritis model. We're working more on a platform extension called Active Targeting, and I will elaborate on that later on in this presentation. And we also now initiated work on combining our platform with oligonucleotides, as we do see that we have also an opportunity there. This is what we do ourselves, and we now have two programs ongoing with two partners, namely Nexogen, where we use their anti-cancer peptides and trap them within our nanoparticles and aim towards enhanced efficacy, as well as that we use a modified version of the polymers and combine it with antigens of PX Biosolutions, an Australian company, and work also towards vaccination application. But let's get started off with the most interesting program, namely CriPec Docetaxel. So to show you some efficacy data in a breast cancer model, so the left figure illustrates the efficacy of Taxotere versus CriPec Docetaxel in equimolar comparison. And what you can see if you monitor the efficacy over time, then if you give only one single injection, you clearly see that Taxotere does have a temporary suppression of the tumor growth, but it relapses fairly fast again. While in our case, we have a really long-term suppression of the tumor growth. Because of the improved safety profile, we did actually a study before this study, namely to determine the MTD. We could demonstrate the much more enhanced safety profile of CriPec Docetaxel in this tumor model. So we were capable of giving a dosage of 125 milligram per kilo. And as you can see, even in case of a fully established tumor, then a single injection was capable of resulting in a full tumor eradication in time. This is all intravenous route of administration, as holds true for the rest of the figures I will demonstrate. So as the concept is the enhanced accumulation of the nanoparticles in the tumor site, we monitor that also in time. And the left figure illustrates the equimolar comparison versus Taxotere. And as you can see, after 2 or 4 days, there is an enormous difference between the amount of CriPec Docetaxel that is still residing in the tumor as compared to Taxotere. If you are capable, as indicated in the former slide, giving a higher dosage, then this difference is even much larger. So really proving the enhanced accumulation of the nanoparticles at the target site, all as the result of the much more improved blood circulation profile, as illustrated in this figure. So the left side illustrates Taxotere, known to be rather rapidly eliminated from the bloodstream, while on the right side you see CriPec Docetaxel PK profile in healthy rats after an intravenous administration. And then I'd particularly like to point your attention to total Docetaxel being the representative of the intact nanoparticles. And as you can see, it's a logarithmic scale. The nanoparticles really have a long blood circulation profile, having a half-life of approximately 16 hours, while there is only a limited amount of Docetaxel diffusing out of the nanoparticles. So you really are capable of bringing much more of the nanoparticles to the tumor, as illustrated in the former slide. So in parallel, what we've done is, in consultation with the Swedish as well as the Dutch regulatory authorities, is done a complete non-clinical safety package, whereby we've evaluated all the different properties, so single acute all the way to the subchronic 6-week dose study, which is the most representative for the actual clinical setting, so giving every 3 weeks an injection. But all of these studies really clearly demonstrate the improved safety profile of CriPec Docetaxel versus Taxotere, as well as also that we took along empty nanoparticles just also to prove that the empty particles, so without the drug, are also proven to be safe, as it's a novel polymer that is tested in this case in a preclinical setting. And it all sums up towards the application of a phase 1/2a study, for which we do have the ethical committee approval already from the Dutch as well as the Belgian sites. We're going to perform the study in Rotterdam as well as in Leuven. And the setup is going to be really very straightforward, namely phase 1 study, which is a dose escalation approach, starting off with 3 patients with a dose, starting dose of 50 milligram per cubic square meter, whereby, maybe for your reference, Taxotere is currently administered 75 to 100 milligram per square meter. And the primary objectives are to measure the tolerability and the MTD of CriPec Docetaxel, as well as measuring the PK profile. This phase 1 study will be followed by a phase 2a study, of which the specifics have not yet been decided on, so I cannot disclose any further details yet. Maybe then to elaborate further on the more platform extension, namely the CriPec Active Targeting, that's what ATN stands for. So what we're working on is actually the area where we've conjugated a targeting moiety to the surface of the nanoparticles. This was already somewhat older work, which we did in collaboration with the Utrecht University. And in this case, we conjugated a EGFR targeting nanobody, so that's the EGA1, conjugated to the surface of CriPec doxorubicin. And as you can see in the survival plot, the enhanced efficacy was clearly detected as compared to non-targeted CriPec doxorubicin nanoparticles. In this case, the nanobody was conjugated via the so-called disulfide bridge, which is known to be not the most stable one in biological setting. So we actually took a step back, went to the lab bench and optimized the chemistry behind it. And what we've developed so far is another approach, namely that we use the regular polymer, and we mix it with a small portion of a polymer that is modified from the surface with an AZAT moiety, thereby generating nanoparticles which present this AZAT moiety on the surface. And then the last step to generate these actively targeted nanoparticles is the combination with a targeting ligand, whereby at the moment we have already successfully conjugated a range of different targeting ligands. And I'll give you an example here of RGD, where we generated nanoparticles fairly small with a nice small polar dispersity parameter, and with a controllable amount of RGD on the surface. We really have by now developed also a method that directly quantifies the number of ligands on the surface of these nanoparticles. With the good entrapment efficiency of the doxorubicin, as well as at the last step, so the conjugation, the click chemistry does not affect the nanoparticle properties whatsoever. What we're currently doing in collaboration with Toine Lammers at the Aachen University is doing an extensive in vitro evaluation, and these are very fresh data showing that if you have RGD on the surface and evaluate the interaction with, in this case, 41 cells, so the integrin positive cell lines, that depending on the percentage of RGD on the surface of the nanoparticles, you do see an enhanced interaction with these cells, whereas that is absent in case of the absence of the integrin receptor on the surface. So this is very preliminary work. We're currently working also on an in vivo study, monitoring the in vivo efficacy as well as in vivo PK profile. So that's work that's still continuing. But I do think in general the CriPec active targeting is a game-changing concept because of the increased interest in targeting different pathways. I think CriPec has a clear added value in this field, as it allows for the entrapment of different drugs within the nanoparticles, as well as by combining it with a ligand or even more ligands on the surface, thereby really aiming for achieving enhanced or even synergistic efficacy. There is, however, one really essential requirement for that, is that we really understand the target biology of a specific indication. And if we know what is required, then we do have the toolbox available that really allows for the rational design of CriPec actively targeted nanoparticles with an enhanced therapeutic performance. And with that, I come to the summary. So Cristal Therapeutics is working on CriPec-based nanomedicine with a clear added value in terms of efficacy as well as safety profile, as I hope I've demonstrated to you currently in the preclinical setting. We are about to start a phase 1 study, and be more than pleased to present those data to you next year. At least I hope I have them then available. And with that, I'd like to thank you for your attention and be more than pleased to take any questions.
Okay, we've reset the clock. So we
Okay.
have 5 minutes of questions now at the end of this session, if we wish to use them. So are there any questions? There's one at the back there.
Thanks for an excellent update. Impressive work. There are two questions. One very technical. How do you put these formulation on the shelf? Is that freeze-dried or in aqueous solution? How do you do that?
At the moment, the material is a liquid solution, but we are indeed working on a freeze-dried product. But at the moment, we do have already a stability of more than a year in the freezer.
Okay. In the freezer, so not in the refrigerator?
No, because of the biodegradability.
Okay, so you probably need lyophilization then.
Right, yeah.
And the second question is a totally different question. I mean, in the phase 1 study, you have a dosing interval of 3 weeks. That is the normal dosing interval for this kind of oncolytics. But in your animal work, you see very clearly that the effect runs for longer periods of time. Did you ever consider to use a different dosing interval?
In the clinic, you mean?
Yeah.
Yes, we have considered indeed, but to get started off somewhere, we thought we first compare with the more traditional approach of the Taxotere. So going for first of every 3 weeks administration.
I just wonder about what the authorities ask for our pharmacokinetics data. You mentioned that you are to measure docetaxel. Was that just a particle bound or also free?
That's a good question. They ask you to monitor the active compound as well as also demonstrate where the nanoparticles themselves are going. So we measured indeed both the still covalently linked material as well as the release material. And then give an insight into the biodistribution over time, so also demonstrating the elimination of the nanoparticles themselves.
If you think about it, can I add a small question? There seemed to be a little wiggly bit at the end of your polymer. Was that anything particularly? The extreme end of the polymer, was there anything on there?
It's polyethylene glycol.
Polyethylene glycol.
Right.
Okay, that's fine. Could I also ask what you think would be the sort of ideal xenograph data to actually translate this project, really? I'm conscious of that in the case of ADCs, you can get 100% survival.
Right.
So can you comment on that, perhaps?
Yeah, I'd say the most, how to say it, the best, though intermediate mice model are the autotopic mice models. So, for example, the implementation in the mammary fat pad. That seems to be the most. Well, of course, it's not really the best representative for the clinical situation, but the most ideal situation for us being a small company going forward.
Okay. Just a follow-on really from the PEG. It's a rarely known fact, but that the osmolality of PEG-containing compounds is a function of temperature. So that if you measure the osmolality at room temperature, at different temperatures, it's different really. So that sometimes makes a difference in the formulation.
Okay, that's something I'll take along. Thanks.
Okay. Any more questions? If not, thank you very much indeed. And we'll move on to the next